Hi, my name is Marirosa Molina, and today I will be talking to you about modeling the impact of urban development in Proctor Creek, Atlanta. We'll be using microbial source tracking markers and pathogen occurrences on these modeling exercises. But before continuing, I would like to acknowledge the collaboration of my coworkers, Blake Snyder, who is located at the US EPA in Region 4 in Athens, and Dr. Mike Sitersky, which works for the EPA ORD lab in Athens as well. For the outline of this talk, I will be talking about the major contaminants that impact urban sources. I will give a description of the Proctor Creek watershed and the sampling and analytical methods that we used. We'll continue discussing the modeling approaches that we applied and which ones were the best models for predicting the fecal indicator concentrations for both traditional, culturable, and molecular methods. We'll finalize talking about the relevant variables that are influencing the concentration of these fecal indicators in the environment, as well as what is the impact on watershed management. Urban systems are impacted by a variety of contaminants. These include nutrients, organic pollutants, antibiotic resistance. Most recently, they have been identified both at the bacteria that are resistant to antibiotics and genes that are harbored in bacteria, and pathogens. These contaminants are oftentimes contributed by the effects of combined sewer overflows, which are present in many of our urban centers across the nation, obviously impacted by the urban runoff that reaches our urban streams, and also by decaying infrastructure. Many times we have sewer line breaks, the sanitary sewer is directly leaking into the systems, into the urban systems, and or we have at times illicit sewer connections into the stormwater drains. So today I will be talking about the study that we conducted in Proctor Creek, Atlanta. This is an urban stream that is about 41 square kilometers. It is located to the west of downtown Atlanta. The watershed is highly developed and mostly at the upper sides, and then less densely developed at the lower sides, or the closer to the pool point of the watershed where it meets the Chattahoochee River. However, there's a variety of land uses that are influencing the presence of contaminants in the system, which include landfills, brownfields, and the railroad tracks and train depot. There are multiple sources of pollution that have created the impairment of the Proctor Creek watershed for fecal coliforms. So it is listed in the 303D list. Now, one question that arises is it the fact that they have this high level of E. coli, this high level of the concentration of E. coli, is it due to anthropogenic sources in fecal contamination from human sources, or could it be due to wildlife? So the research objectives for our project were to develop models for the concentration of E. coli and a human-associated marker in this type of urban watersheds, and identify and compare the variables that mostly influence the presence of the anthropogenic contamination in the system. The methods included collecting grab samples from November 2015 through July of 2017 on a biweekly basis at 12 sites. We conducted analysis for determining the concentration of E. coli at the culturable level using the defined-substrate methods, coli-18, and using a molecular qPCR approach. Then we analyze for the human-associated marker HF183, is a bacteroides marker, and identify the presence or the frequency of the presence of Salmonella and Shiga-toxin. Shiga-toxin is found in organisms such as E. coli, but the enterohemorrhagic E. coli. In addition to the microbial contaminants, we perform standard water quality parameter analysis. We measure temperature, dissolved oxygen, pH, and specific conductance and turbidity. In this first graph, I am showing the log transform E. coli concentrations for dry and wet periods in the main stem and tributary sites of the Proctor Creek watershed. You could see basically in this, we split the sites. We had 12 sites, and about half of them were on the main stem and half of them were on tributaries. We conducted the analysis and based on the dry and wet periods over the watershed, and determined that the concentration of E. coli was significantly higher during wet periods at several locations throughout the main stem. But one important aspect of this graph is the fact that throughout our study, our numbers were the concentration of E. coli was found to exceed the EPA E. coli recreational water quality criteria throughout the year. In this second graph, I am showing the concentration of the human-associated marker HF183, as again for wet and dry periods in the main stem and the tributary sites as before. The concentration, it was found that the concentration of the HF183 was significantly higher during the wet periods, again, both in main stem and in various locations of various of the tributaries that were impacting the main stem. But of notice is the concentration that was identified from at the Greens Ferry location. The Greens Ferry is a decommissioned CSO, so it is not online anymore. However, consistently it produced the highest concentration of the human marker, and it was certainly higher, significantly higher during the wet periods. In addition, in the main stem, we observed the highest concentrations at the North Avenue point, and this is another CSO that is present in the city of Atlanta, but now this CSO is currently active, and the concentration was as well higher during the wet period. In these next figures, I am indicating the frequency of the Salmonella and Shiga-toxin across the watershed throughout our sampling time. The top panel indicates the presence of Salmonella. The bottom panel indicates the presence of the Shiga-toxin. And this is a land use map of the watershed, and you could see the intense red color indicates the highly the more densely populated areas. So for the Salmonella, you can actually see that it was detected more often in the lower watershed, in the lower sites, which are the ones closer to the pour point of the watershed, as it is meeting, as Proctor Creek is meeting the Chattahoochee River. However, the Stx-2 was widespread all across the watershed, and it had higher concentrations both at the upper and lower sites. Next, I would like to discuss the application of the virtual beach tools, or the modeling approaches that we took to analyze the data that we gathered from the watershed. We, first of all, the virtual beach statistical tool is package developed by the EPA. It contains a variety of statistical tools for people to perform predictive modeling. We selected the multilinear regression analysis, just fitting a linear equation to the observed data. In this case, we used environmental parameters that we're measuring to predict the concentration of our microbial targets. And we also, in comparison, in addition, we use the generalized boosted regression models, or GBM, which is basically a linked chain of simple decision trees that keeps adding the environmental parameters that were measured until it finds which ones are the best combination of parameters to explain the variability in the target. This graph demonstrates the results from our modeling approaches and compares the results that we obtain from the GBM versus the MLR. With the targets that I discussed previously included the culturable E. coli and the qPCR E. coli, as well as the human marker. So we were trying to use environmental parameters that would explain the variability of these signals in the watershed. It is to notice that the human marker, the HF183, was modeled using the influence of both the culturable E. coli for the first column and the influence of the qPCR E. coli on the signal. So basically we wanted to determine what is the effect of, or the relationship of the culturable and qPCR E. coli on the presence of the human marker in the system. So the results indicate that the GBM is far better than the MLR based on the general statistics, but particularly the R-square. So we obtain an R-square of 0.6 to 0.8 with GBM across the different targets. In this next table, I am presenting the influence and the percentage of the influence of each independent variable that got picked up by the GBM model, since they were the better models to perform. And so you could, it is relevant to see that for both E. coli, the molecular and culturable, the percent of forest coverage has about 20% of influence on the model, and followed by pH and turbidity for both types of indicators. In the case of the HF183, the human marker, we have that the culturable E. coli explained about 42%, had about 42% influence on the model, and while the qPCR had almost 50% influence on the model, meaning that there's a high correlation between the E. coli and the human marker, and potentially indicating that the sources of the E. coli in the system is actually from human sources. The other variable that was important for the HF183 was the sulfoxygen, followed again by the percent of forest coverage. In the case of Salmonella and Stx-2, we use a generalized linear model approach. In this case, we had to use it because Salmonella and Stx were both enriched, and we were using the presence-absence calculations rather than a concentration. But we found that we were able to predict Salmonella with 66% accuracy using temperature, pH, the rain intensity on the 48 hours prior to sampling, and the impervious coverage. The Stx-2 was predicted about with a 52% accuracy, so a lot less, and actually the variables were not found to be significant. So in summary, the results predicted that both culturable and qPCR E. coli and the human marker HF183 were able to be predicted with high accuracy using the GBM. The percent forest was the most influential watershed variable predicting E. coli, and the second most influential predicting HF183. Actually, this percent forest, we look at the coefficients that we obtain for the multilinear regressions and determine that it has an inverse relationship to the markers. Therefore, the lower the percent coverage or the percent of forested land, the higher the concentration of these markers in the environment. Salmonella was predicted at a 66% accuracy using temperature, pH, rain intensity, and the impervious coverage. And finally, the Shiga-toxin was identified at the headwaters of the watershed, but an accurate model was, we were not able to produce an accurate model. And in conclusion, we have that this decision tree analysis, or GBM, seems to be an effective way of modeling the concentration of the fecal indicators in a watershed, and it seems to be more effective than the multilinear regression approach. The modeling demonstrated the relationship between E. coli and the human-associated marker, indicating then that the E. coli, that the high numbers of E. coli that we have in the watershed are very likely being contributed by human sources. And managing forest coverage may help reduce the presence of these fecal indicator bacteria, microbial source tracking markers, and pathogens, thus improving the water quality in urban ecosystems. Thank you for your attention.